Well, good morning and good afternoon. I'm Gael Forter, the Chief Commercial Officer of Volition. It's a pleasure to welcome you to this webinar today and to introduce you to Dr. Jace Mikalev, our Chief Scientific Officer, and Dr. Andrew Retter for their presentations on the breakthrough cancer detection method. Before the detailed science, a few words. Our mission is to save lives and improve outcomes for millions of people and animals worldwide. To do so, we're focused on developing a simple, easy to use, cost-effective blood test to help diagnose and monitor a range of life-altering disease, including cancer, or original focus. Finding cancer, and especially finding it early, has often been described as looking for a needle in a haystack. And indeed, as Jake will explain, it is a needle in a haystack when the needle is also made of hay. The new method presented at ESMO earlier this week demonstrates we have a way to find the needle. This is an incredible breakthrough, and I know that you will be interested in the presentation from both Jake and Andy. The market opportunity is big. It is very big, in the billions. It is bigger than us, which leads us to a commercial strategy. We will license this technology early and seek upfront payments, milestone payments, royalties, and sales of key components. While the data presented is literally off the press, we're already in discussions with interested parties. With that said, I'll keep it there for the commercial part. I will now pass over to Jake and Andy to share more details. Thank you.
Now you've heard from Gael about the commercial opportunities, I'm going to tell you a little bit about the science behind our breakthrough in cancer detection. First of all, a little bit of background. On this slide you can see the structure of chromatin, and we've shown this picture many times before. It's a string of nucleosomes held together by a string of DNA. But what we've added to it in this picture is a complex called CTCF. And CTCF is a transcription factor that also binds to DNA like a nucleosome, but has completely different functions. CTCF is actually a very important protein that regulates what proteins are made in the cell and actually regulates the structure of chromosomes within the nucleus. There are many big hospitals and quite a few companies that are involved in performing liquid biopsy ctDNA assays for circulating tumor DNA. The challenge that these companies and hospitals face is that most of the DNA circulating in a person's blood, even if they have cancer, is normal, especially at early stage cancer. In stage 1 cancer, for example, it's likely that only 1 part in 1,000 or 1 part in 10,000 of the DNA that's circulating comes from the tumor. The rest is normal. This really is a needle in a haystack problem. And what makes the problem even more difficult is that the needle is really made of hay, because the tumor DNA is not chemically any different from any of the normal DNA. And of course, because it's in the same person, it has a near identical sequence. So how do current ctDNA assays work? They've all really got the same mechanism and the same method, and it hasn't actually changed since the inception of liquid biopsy 10 or 15 years ago. What happens is that you take a blood sample, you extract all the DNA from that blood sample, that is the normal DNA and the cancer DNA, if there is any. You then amplify that DNA to make more, and you do what's called a library preparation, which is basically getting the DNA ready for next generation sequencing. You then do the next generation sequencing. That's a very expensive part. But even when you've got all the sequence data for all of the DNA that was present in that blood sample, it's still mixed up. It's still only 1 part in 1,000 or 1 part in 10,000 from the cancer. The problem's very difficult, and so it's solved by putting the whole sequence data into a big computer program which have very sophisticated bioinformatic algorithms to decide whether or not any of that DNA actually did come from a tumor or not. And over the years, the results for liquid biopsy have improved. They've improved a lot. And the reason for that is that each of those stages has actually improved. So the bioinformatics is better, the sequencing is better, the library preparations are better, and so on. But the basic method is unchanged in a decade or more. The reason the basic method for liquid biopsy is unchanged is because there isn't really anything you can do with DNA other than sequence it. The real answer would be to physically separate the DNA that comes from the cancer, and then detecting it would be as simple as a COVID test. A COVID is a PCR test, and this would be a PCR test. That would make for a very simple ctDNA test. As the acronym says, let's keep it simple, stupid. Having said that it would be good to separate tumor-derived DNA from normal background DNA, of course, it's impossible to do that chemically. However, DNA per se is not what circulates in the bloodstream. What circulates is pieces of chromosomes. So the DNA circulates as protein DNA complexes. Mostly those are nucleosomes, but there are many others as well. One of the others, as you can see on this slide, is CTCF. What we have found is that there are combinations of DNA sequences that occur on CTCF proteins only in cancer. So if you separate out all of the CTCF DNA fragments and then do a PCR, you will detect whether or not any of the cancer combination involving both CTCF and a particular sequence is present. And that will tell you if a person has cancer. What this technology allows us to do is a completely new method for the analysis of ctDNA. For the first time ever, the sequence of events is not simply to extract all the DNA, amplify library prep, sequencing, and bioinformatics. Instead, we will pull out all of the CTCF molecules and their associated DNA fragments and do a simple PCR test to identify whether or not any of them come from cancer. Scientifically, what makes that possible is really quite simple. CTCF binds to something like 60, 70, or even 100,000 places in the human genome, and it binds to different places when you have cancer. So when a cell becomes cancerous, CTCF binding is lost in some places, but it's also gained in many places. And so where there is a gain of occupancy in cancer, those sequences will not occur on CTCF in a healthy person, but they will occur in a person with cancer. And in a healthy person instead, all those sequences will occur on nucleosomes. So what does that look like in practice? On this slide, we've got nucleosomes and CTCF molecules, and they are covered by DNA molecules, and most of those are healthy. We have some that are covered by the yellow DNA molecules, which contain the CTCF binding sequence. A lot of those will be on nucleosomes, but in cancer, some of them will also be on CTCF molecules, and you can see that in this picture. Using the occupancy properties of CTCF, we can select sequences that only occur on CTCF in cancer patients. And in this slide, you can see that in a healthy person, the yellow DNA sequence, which represents the cancer-associated sequence of interest, occurs on nucleosomes, the green circle. In a person with cancer, they also have nucleosomes with yellow cancer-associated DNA attached, but in addition, they have the yellow sequence attached to CTCF, and that does not occur in the healthy persons. But if we can remove all of the nucleosomes by separating out the CTCF, then we're left with this picture. So now we have a mixture of CTCF proteins bound to DNA sequences, some of which are yellow and come from the cancer, and some of which are still healthy. Now we can extract the DNA, so we're left with just these pieces of DNA and the CTCF is gone. If you have cancer, you would have the yellow sequences. If you're healthy, you would have no yellow sequences because they would only have occurred on nucleosomes and they would wash away. So the presence of the yellow DNA sequence is an indicator that you have cancer. And we can test for this by a simple PCR, which is like a COVID test. What this means is that for the first time ever, we can physically isolate ctDNA cancer-associated fragments from the normal DNA sequences that contain the same sequence. There's an overall picture here of the method. We take a plasma sample and we add an anti-CTCF antibody to that. That's the purple Y-shaped thing. It pulls the CTCF out of the plasma. We then wash all of the nucleosomes away and we're left with an antibody bound to CTCF. We then extract the DNA from that CTCF antibody and we do a simple PCR test to see whether or not the yellow cancer-associated DNA fragment is present. In fact, one of the beauties of this technology is that biomarker discovery is rapid and very straightforward. So what we did was to take some healthy blood samples. Some samples were from people with other diseases like inflammatory diseases, and some blood samples from people with cancer. We then did the same procedure as before. We pulled out the CTCF associated DNA fragments, but this time we did sequence them for biomarker discovery. So what we did was to see what sequences were present in the cancer samples that were not present in the other samples. And we found 29 sequences that met our criteria. We completed it fairly recently, and then we ran a very preliminary clinical proof of concept. As I said before, we found 29 biomarkers that were interesting. We actually developed PCR tests for 10 of those, and now I'm going to show you just how well those 10 tests performed in reality. Some biomarkers were quite specific for leukemia, but most of the biomarkers were also effective for the identification of a variety of common solid cancers. And different biomarkers were effective for the detection of different combinations of solid cancers, including at early stage cancer. This means it's possible that we will be able to produce assays that can detect particular cancers, for example, a breast cancer test, a prostate cancer test, and so on. And we're currently developing biomarkers for the common solid cancers, including breast, prostate, colorectal cancer, lung, and liver cancer. As I said earlier, biomarker discovery is rapid and low cost, so this won't take us a great deal of time. One point I would like to reiterate is that CTCF has something up to 100,000 or so binding site loci in the human genome, and this represents a potential whole new class of hitherto unappreciated biomarkers that we can use for the detection of cancer. How does it work in practice with real patient samples? So first of all, just using one single PCR test, so now it really is like a COVID test, we were able to detect 61% of leukemia cases with 98% specificity, so really quite high specificity. And you can see that in the ROC curve here, and you can see it pictured with the actual results on the right, where pink is a positive. So we've got one false positive amongst the healthy samples on the left, but lots of pink true positives amongst the leukemia samples on the right. One thing I do want to emphasize is that this is exactly the same as a glucose or a urea or a cholesterol test. There's a simple cutoff. If you're below the cutoff, you're negative, and if you're above the cutoff, you're positive. You don't need a computer. If we now add a second PCR, so we've got a panel of 2 PCR tests, then the sensitivity goes up to 74% of leukemia cases detected, with a very slightly lower specificity at 96%. Most of the PCR tests also detected different solid cancers, and there were 2 or 3 of the assays that were particularly good for detection of colorectal cancer. And in fact, if you put those 2 assays together, we can make a 2 PCR panel that detected 77% of colorectal cancer cases at 92% specificity, and that represents the black dot on this diagram. So what we've achieved here is a blood test that gives nearly as good results as a fecal test, but of course it's much easier to use, and that wasn't even developed on CRC. It was developed for leukemia. We're now developing individual biomarkers that are specific for CRC, and we expect to get results that exceed those that you can get with FIT. And of course, we're developing other tests for lung, prostate, breast, and so on. There's a lot of talk in the liquid biopsy field about multi-cancer early detection tests. So we put another panel together of 2 PCRs that were not specific for any particular cancer or specific for different ones, so that we could combine detection of multiple cancers. And we looked at colorectal, breast, prostate, and liver cancer. We've got patients for 10 of each, except for CRC, we had 13. And we've got positive results of between 50 and 70% for the different cancers. However, what I really want to highlight is cancer stages, of course, because detection of a stage 4 cancer doesn't really help the patient a great deal. What would really make the difference is telling a person that detecting a person's cancer when it's at stage 1 and stage 2, and something can be really done to help them. So we did detect a lot of stage 4 cancers, 75% of them, but the really astonishing thing is that we can detect between 1/3 and 1/2 of stage 1 and 2 cancers. And that really is something I think that nobody has achieved before, to detect early stage cancer in a simple blood test that is quick and low cost. Finally, I'd like to reiterate the advantages of physically separating tumor DNA from normal DNA, because it leads to a much simpler, lower cost, better test. First of all, the cost of traditional NGS assays are $1,000 or more, whereas this is going to cost much less, probably in the region of $100 or so. The traditional assays are very complex, they're not automated, whereas this can be automated. I think the big IVD firms can make a machine that will do this at the press of a button. Traditional NGS is really suitable for high-tech labs. The NHS in England, for example, has 7 labs that perform all of the tests for the whole country, whereas this assay can be done in any central hospital lab, especially if it's automated. It's a lot faster, it takes less than a day, even if it's manual, whereas traditional assays take days or weeks. And of course, there's no library preparation, there's no sequencing, there's no computer involved. And the answer is a simple plus-minus answer, like a COVID test. You don't need a computer to analyze the result. What's coming next? Well, first of all, we will be publishing a paper on this method, including all the details of how to do it and so on. And then we will be next developing particular tests for breast cancer, prostate cancer, lung, colorectal, and so on. And details of those will be coming out over the next few months. As Gail said at the beginning, the mission of our company is to produce life-saving diagnostic tests, and that's also my personal passion, especially if they can be low cost and accessible to everybody, not just accessible in rich, developed countries. Having said that, I'd like to pass you over to somebody that actually understands medicine, and that's Dr. Andrew Retter.
Hello everybody, I'm Dr. Retter. I am a consultant to Volition, and I'm very excited to take you through this talk on cancer today. And thank you to Jake for talking just now. Just to be clear, I'm a consultant to Volition. I do work in the National Health Service, but for clarity, the views expressed here are my own and don't represent those of the NHS Trust where I work. Cancer is the second leading cause of death worldwide. over the next 20 years, we expect the rates of cancer to go up by about 1.5 times, leaving a huge burden of disease in the population. 5-year survival rates are improving for cancer, which is a brilliant thing. And there's a huge amount of research and the field is rapidly growing and changing. But about 45% of cancers in the UK are still diagnosed in a late stage, in stage 3 and stage 4, where treatment options are more limited. The treatments tend to be much more invasive, much more prolonged, and indeed perhaps even more severe and more difficult for patients to tolerate. And many patients will be older and just too frail to tolerate the intensity of those therapies. So there's a key critical need to improve the diagnosis of cancer and the stages at which we can diagnose cancer. So everyone understands that the earlier you treat something, the easier it is to treat, to interdict, to change its course, and hopefully cure more people. How might the new technology from Volition help? It's aligned with Volition's key principles of diagnostic enrichment. This is using and leveraging modern pioneering biotechnology to improve early stage diagnosis, to monitor diseases and to review their progression going forward. If we can diagnose patients earlier and monitor their treatment more efficiently, perhaps reducing the need for expensive, costly CT scans and MRI scans, or even positron emission scans too, hopefully we can lead to faster, more rapid diagnosis and identification of problems or progression when they occur. What's coming next in this area? Well, we're already in the advanced stages of finishing our first paper discussing transcription factors and their applicability in a large number of cancers. We're starting to develop specific biomarkers for lung, colorectal cancer, prostate cancer, breast cancer, and liver cancers. And together, those are the most common malignancies. Abstracts and more posters are going to be presented at a number of specific or cancer-specific conferences throughout 2024. We have ongoing work with a number of key opinion leaders from centres of excellence to accelerate and progress that research further. So in summary, we're extremely excited to announce this novel breakthrough technology, and we're extremely excited by the transformative effect it could bring to so many patients. It has huge potential of significant clinical and commercial benefit, and it contains a number of truly world firsts. Thank you very much for listening, and I'm delighted to hand you back to Sue. Thank you.